Thank you very much. And before I start, I think I would like to reflect on who's contributed to this effort, and Antonio. So I guess it's the first time the three of us have been together presenting this, which is a nice thing. And I also have to say that they introduced me. It was almost like a chance. I came across the group in Madrid. I mean, I'm from Spain, but I'm in England, so you feel a bit like an expat. So Antonio introduced me not only to this whole world of stochastic processes in a very theoretical way, but then I guess he made possible for Martin to come to our group. And I guess it's been a lovely adventure. So, and I think it's also time to say thank you to Jesus, who's not here with us anymore, Jesus Arzalejo, so, because he really pushed this. So what's the story that I would like to say? Well, let me get started, because how many of you know some immunology, just so that I get a sense of, right, okay, so what is the question that I would like to address? So as we know, T cells, before they move to the periphery, they originate from the thymus. They come from the bone marrow. So some cells that are precursors of T cells come to the bone marrow. So come from the bone marrow to the thymus. And this is sort of a picture of a cross-section of the thymus. There's some structure here, but to me what I would like to point out is that the crucial events are T cells, round cells, talking to, dendritic shaped cells, and those interactions shape early on the fate of these cells to be selected. So what is actually the process in the thymus? So the process in the thymus is, as I've said, now this is sort of a different side view. You've got the cortex and the medulla of the thymus. They come, so from the bone marrow, they actually enter through precisely this junction between these two regions. And these progenitors that are called different names, it doesn't really matter really for the purpose of this talk, they go through these stages, DN1, DN2, DN3, different maturation stages, and then they eventually move to the medulla. But basically, every step along the way from a certain point is mediated by interactions between a T cell, a T cell 2B, that is presenting on its surface the T cell receptor with a certain type of antigen-presenting cells, and those interactions mediate this selection process in the thymus. In the thymus, of course, you need to check for functionality, that the T cell is working, but also that the T cell signal is not too large, because that could be selecting for thymocytes, precursors of T cells that could give rise to autoimmune disease. To me, this is a beautiful, I still remember the day when I was exposed to this, and I was quite old. But it was, as a physicist, it was such a beautiful exercise of how biology is sort of helping us to be protected. We need to live very long in a world of pathogens. So there's a beautiful development in the thymus of these thymocytes, both in space and time. And this hope for a given cell, for a given thymocyte, at most they've got 2 weeks to sort of, do I make it or I don't? So back to then this question, this is an exquisitely stringent test. And I always, I know it's a very bad joke because nobody likes exams. But if someone tells you, well, this was Antonio and Martin knows that in Spain, pretty much that's the 5% chance to pass exams in university, at least when I was there. So, it's not that the drill is not familiar, but if someone tells you, you have a 5% chance, it's not zero, but it's not great. So this is the fate of these thymocytes. And the thymocytes, as I was saying, they talk to special cells that present a type of ligand that can bind to the T cell receptor on the surface of the T cells to be. And of course, just imagine, if you need to be a radio, and you cannot detect waves, if you cannot generate a signal, then, you die by a type of death called death by neglect. If the signal you receive is too strong, that then you die by apoptosis, and that's called negative selection in the sense that you could lead to autoimmunity. And if you are, which is interesting, if you're too bad or too good, we get rid of you. If you are, intermediate, we keep you. Export to the periphery, and periphery in this context is the blood and all the lymph nodes in our body, and that's called positive selection. So the idea is that in this pool of different T cell receptors, you select for the ones that are going to be intermediate. And I should mention that the selection is mediated by bits of the proteins that come from household proteins, so peptides. So the idea is that that's why you want to have intermediate ranges for your own proteins. So before I move into the mathematical model, I think it's important to ask ourselves why we're doing this, but also what is the immunological evidence today, but at the same time, what is the immunological evidence that I take to build the model, because of course I can't take the whole complexity of. So basically I like to have a slide like this so that you know what we're putting in. And also one advantage or one reason why we do some maths here is because we believe that at least we are being extremely honest. In mathematics or in physics, you can't define what you put into the system, and then you can a posteriori say, well, am I getting interesting results, or am I only getting what I'm putting in, which sometimes, we can do very well, and then pretend we're doing some magic tricks. So what is it that we know? What's immunological evidence? Immunological evidence is that, of course, in order to have a proper adaptive T-cell response, we need to be able to have a T-cell repertoire of cells that can respond to any potential pathogen that I may be exposed in my lifetime. And of course, our bodies cannot predict the precise nature of the pathogenic encounters that I have in our future. So what you have to do is, once you create, through this very expensive process, which is thymic selection, I always ask, Sarah, I guess it's the one immunologist I know here, and I always ask myself the question, how expensive is, metabolically speaking, the thymic production, if we could compare to other metabolic processes? And I have a feeling that it's a very expensive process. I'm just guessing, but to me, it's an expensive process, thymic selection. So how can then we, given that we're creating these cells, sending them to the periphery, blood and lymph nodes, how do we maintain them? Because they're very valuable, right? We live very long lives in this world of pathogens. So what is it that we know about this homeostatic regulation? So we have, and I'm only talking about naive T cells, of the order, okay, what is a naive T cell? Is a cell that has come from the thymus and hasn't seen its specific antigen, right? So, of the order of 10 to the 11 cells, naive, that are distributed in different families. And these families are sort of defined by the nature, the molecular structure of the T cell receptor that they express. So every naive T cell expresses on average 30,000 copies of these T cell receptors. So every cell that has the same molecular structure, you put it in what we define a clonotype. So, you have these families and you can do the division. So of the order of 1,000 cells, that doesn't mean that all clones are the same size, right? But roughly speaking, you've got for each family about 1,000 cells, which, were rather big. So 1,000 cells is not a lot, and we've got lots of lymph nodes. So how do I maintain this population of cells while I am alive or still young? So the idea, and this is the immunological evidence, is that in order for these cells to survive, they need to get a bit of a tickle from professional antigen-presenting cells. And I'll explain this in a... So the idea is that you get a bit of a tickle to just have not an immune response. This is only homeostatic proliferation, homeostatic in the sense that I'm maintaining, it's not an immune response. And in this way, by having this bit of a tickle, I guarantee coexistence and persistence of all these different families that are going to be important because they're going to cover the broad spectrum of pathogens that I can be exposed in my lifetime. And of course, the idea is that I've got different clonotypes of different cellular sizes. And by cellular size, I mean the number of cells in a given family. Unfortunately, as we know, some of us here in the audience know, as we age, we decline. Our physical ability declines, not our mental, right? Wishful thinking, but So, unfortunately, this decline is both in size and diversity. So, this is the hallmark of the aging process. We have fewer cells, so not only this number goes down, but also the diversity, how many different families we've got. So mathematically, losing something is extinction or absorption. So the idea that, I mean, this was, and I should also say that I was introduced to all this by Hugo van den Berg. I'll give him acknowledgements at the end of my talk. But then the idea is that we started 2008, that's when we started thinking about these models. And then about 4 years ago with Antonio and Martin. So that's where we are. So the idea is I'm going to guide you to how we can make a model of naive T-cell homeostasis that has extinction. So just to ensure that everybody's on the same page, what is an antigen-presenting cell? So it's a cell that on the surface has an MHC molecule with a little bit of a peptide. For the purposes of this talk, these peptides will all come from household proteins. Of course, what we know if you have an immune response is because you've got also antigen-presenting cells that in this sea of peptide MHC complexes on the surface, and each antigen-presenting cell will have of the order of 10,000 of these units. A few will have the red peptide. I don't know why we always like red with danger. Isn't that funny? I don't know. Maybe purple. I don't know. Just right. But the idea is that, but as I've said, for this talk, we'll be here. Our antigen-presenting cells will always be there. So this would be an example of something you see in the thymus or a homeostasis, and for an immune response, what you see is antigen-presenting cells with a few, less than 100, when you've got sorry, did I say 10,000? No, I meant 100,000 peptide MHC molecules on the surface. If now I look at the T cells, what is it that we've got? So T cells on their surface have T cell receptors, and that's what makes them a T cell. No other cell in our bodies will express the T cell receptor without being a T cell. And they express multiple copies of the same molecular structure. So that's why I make them all the same color. So this is clonotype 1, 2, and 3, of the order, as I say, 30,000 copies. And what we know in humans, that the diversity, the number of different clones, is of the order of 10 to the 7, 10 to the 8. And I think this is very interesting. The nature of these TCRs, molecular structure, that is actually generated randomly by something called genetic recombination. The idea is that for a virus, it would be pretty bad to have a sense of which TCR structures you can generate. So that's just a random, so that you get lots of different T cell receptors. This is this diversity. And of course, if you do something randomly and not talking to the peptide presentation, it's going, no doubt, these T cell receptors are going to be able to recognize self-peptides and generate autoimmune responses. And important is also that from a recognition point of view, there's not just one single peptide will be recognized by a single TCR. It's not the old-fashioned view of lock and key type of thing, where the one key fits the one lock. There's cross-reactivity and promiscuity in these interactions. But the idea is that you generate in the thymus this potential autoimmune TCR. So this is the picture now that I want to go getting into the model. So just imagine, I know I still say this, but it's interesting that 10 years ago, that was sort of what we wanted to have, and slowly we're able to have immunologists telling us where we can go. So what is it that I want to say here? So if, as a mathematician, well, as a physicist really, if you want to understand this, what you would like your immunologist to say is, okay, if you put all the potential antigen-presenting cells on the left-hand side and all my different clonotypes that I know, because I know the gene segments that can give rise to the T cell receptors, can you tell me which, right? Let's suppose my favorite clonotype is T1, but I want to know if a certain presenting cell can talk to my T cell, which other families can it talk to, right? And this can be done for the different antigen-presenting cells, and the idea is that you get a bipartite graph of this type, and if we could get this, that would be very nice. Unfortunately, of course, this is a very challenging experiment. Yeah, measuring these things, it's a bit of a headache, but slowly we're getting there. And I would like to mention here the efforts of Paul Thomas to try to quantify cross-reactivity, for example, in the context of flu peptides. You know, how many different T-cell receptors can be linked to different epitopes of flu, for example. And now also, I don't know if you're familiar with tetramers. Tetramers can fish specific T cells, and if you have tetramer specific for a certain flu or virus epitope, whatever your favorite infection is, you can fish this from your own blood. So slowly, we're getting there to get this picture. Right, so now we move to what can we do with pencil and paper, because that's what we wanted to do. I mean, I really like computers with some, you know, within reasonable limits, but I have to say I like my pencil and paper and eraser more. So if we wanted, I mean, to me the question is, why did we decide to go this way and not a deterministic way? And I think the main reason is, if you want to understand extinction events, deterministic approaches aren't going to do the job for you. That was pretty much the way. Not because we want to be fancy. I think fancy is a bit silly, right? Sometimes you need to be fancy, but there are times where you need to really think about what is the best that I can do to explain what I see. So this is where we were. We convinced ourselves, if extinction is what we want to model, then let's think about a stochastic model. So what is it that we would like to describe? The number of T cells of a given clonotype. So the variable that I would call x, let's imagine I've got a time t equals 0, which is the initial time. Let's imagine it's a time when this thymocyte is ready to export into the blood. So I can have of a certain family 0 cells, or 1 or 2 or 3, and x of t can take any number of natural numbers and 0. I never know if 0 is natural or not, you know, but let's imagine it is. And from a perspective of defining my stochastic, we're going to assume that it's stochastic and Markov. All I need is, if I know how many cells I've got at time t, let's imagine I've got n, in a little time interval delta t, what's the probability that I've got m cells? So this is called the transition probabilities from n to m. And what is it that we are going to assume these cells can do? So the immunological evidence tells us cells don't live forever. Nothing lives forever, really. So if I have n cells, there's a chance that I lose 1 cell due to death and I go to n minus 1. These cells are maintained by homeostatic proliferation, a little bit of a tickle from an antigen-presenting cell. That means I've got 1 division. So if I have n cells and I've got 1 division, I move to my state, n plus 1. And of course, let's imagine, you know, I start with n and there's some deaths, I can go to 1. So if I have 1 cell of a given clonotype, of course, if that cell dies, then I land in 0. And what I didn't say, and I should have said, is that the thymus, as we age, it involutes. So thymic export, pretty much, for someone like me, it's 0. Therefore, if I run out of cells of a certain clonotype, those are not coming back. And therefore, I'm stuck here. So these are the extinction events and the loss of diversity, because if I lose the clonotype, then my clonotype diversity goes down by 1. Is that clear? Notice that this birth rate from 0 is 0. So if I am in 1 and I land in 0, I can't get out of 0. That is the definition of an absorbing state. And of course, the probability of state 0 can only grow in time. Yep, increase in time. So what is the model? So I would explain it a little bit better. So again, on the left-hand side, what I have here is the potential self-peptides coming from our household proteins that my immunologist is going to give me. And on the right-hand side, I have the different families of clonotypes. So what is it that I will need? So for a certain, and I'll label Q, the peptides, and I, the clonotype. So let's imagine I've got a given peptide, my immunologist tells me it can give homeostatic signals to those clonotypes. So that will be called, this set will be called CQ. Yep. Then I can choose my favorite clonotype, call that I. And again, my immunologist tells me, I look back, I receive signals from this peptide, so this set is called QI. So the idea would be, for a given Q, I know all the CQs, and for a given I know all the Qs, and I can then, so let's imagine, for example, that's important, so let's imagine that's what my immunologist tells me. My immunologist tells me this bipartite network has this structure, so I think important to point out here, and this is no surprise to anybody in this audience, I really believe, is that CQ, the cardinality of this set, how many cells I've got in this set, the number of T cells that receive a signal from Q, if I is in that set, will be how many cells there are in Ni, plus what we define as NIQ, which is the competitors. Just imagine, we're going to be egocentric, I is our favorite clonotype, and we want to understand how this clonotype changes in time. So if Q belongs to if Q belongs to QI, then I want to see what are the competitors for that peptide. That's the idea. So with that, of course, let's imagine that my immunologist tells me QI is these 3 peptides. So let's go one at a time. So for that peptide, 3 peptides. So for the first peptide, there are 2 clonotypes that receive a signal, I and something else. So that is the competitor. Yes? So for this other peptide, there's no competitors. And for the third peptide, I've got 2 competitors. So the idea is, what can you do? So as it's clear from the picture, having competitors implies that in order to understand the change, the dynamics of the number of cells in this clonotype, I need to know also, or I need to have equations to quantify changes in the number of cells of those clonotypes. But I don't want to do that. I said we really want it to be pencil and paper. So what is it that you can do? For the purposes of this talk, I will not go through the derivation. If anybody's interested, I'll be delighted to walk you through that. But the idea is you can do something called a mean field approximation. So I'll explain it here. What is that? Okay, what we assume is any of the competitors, any of the cells that I've labeled cyan in this diagram, they all have, for the purposes of my approximation, the same number of cells that I'm going to write as average n. Just imagine you can do that. I mean, the question is this a good approximation? And a posteriori, can we check that it's a good approximation? So the answer is yes. There are regimes in which this is a good approximation. And the good news is, immunologically speaking, this is a good approximation because for a given clonotype, the number of competitors is very large. So you can treat sort of the competitors as a sea of competitors, and all you need to worry about is for a given clonotype, for a given peptide, how many competitors you've got, and all of them with a certain size average n. So that's the idea. So under that approximation, which that's why I put my little star here, that's the mean field approximation, I don't have to define a multivariate process with lots of different variables all coupled together. All I need to understand is a one-dimensional continuous time birth and death process, which is the one that I anticipated. So I move up and down n plus 1, n minus 1. I only have death events or birth events. Deaths, we're going to assume, natural death, that all cells of a given clonotype die with the same rate, and that's called mu. And one can actually show, and I'll explain what this equation means, that the rate, if I am in state n, and one of my cells divides by homeostatic proliferation, that rate is given by this expression. So how do you digest that? Because I also have to tell you, when we derive this, we said we are stuck. I mean, like what do we do here? So what is this? So phi, let me explain all the parameters. So for death, again, natural death, linear death, all cells of a certain clonotype have the same rate to die, and we call that mu. Notice that this is a parameter that characterizes the clonotype. Phi is the proliferation rate of the T cells of this clonotype. So given the environment of antigen-presenting cells that can signal to them, that is the rate at which they will divide. Nu, which comes from this approximation I've told you, is the number of competing clonotypes for the clonotype under study. So nu, if nu is very small, if nu is 0, that means you don't have any competitors. If nu is 1, you have 1. If nu is very large, you're competing for many. And average n is what I told you is the characteristic size, the number of cells that are in competing clonotypes. And what you see here, this R, that sum is summing over what we call the competition index. So how many competitors you've got for a given peptide that gives a signal. So summing over this, the idea is that when you look at this diagram, for example, it doesn't matter if this, for example, let's imagine that Q1 and Q3 were shared, were seen by I, but also by 2. Let's imagine I had another arrow here. So all that matters is if Q1 and Q3 are shared by 2 other competitors, for the purposes of this approximation, those 2 peptides would be the same, and R would be 2 competitors in this term. So that's the idea. So what you see is that you don't get the full signal if you had 0 competitors, but you have to divide by the number of cells in your clone, n, and the number of competitors. So you're sharing the signal with your competitors, as it's the case when you are sharing resources. So I would point out again, this was derived by my first sort of official and my PhD student, Emily Sterk, who was wonderful, and 3 parameters that describe the clonotype, and 1 that is not a property of the clonotype, but of this sea of competitors. So what is it that we showed? And this was the paper, sorry, God, no, wrong. I hate, I'm terrible with these things. So this paper, 2008, I mean, that's how long we've been in this game. So Emily with Hugo basically drove all this. So what is it that we're able to show? So something that you do, and I have to say, back when we got this, when we were able to obtain this, we thought, God, all we can do now is numerics, which for me is a disaster. I don't want to be there because I feel very vulnerable. I don't know about you, but I really feel like... So beautifully, and I have to say, if you want to learn about stochastic processes in biology, the best book to start is Linda Allen, stochastic models, stochastic processes in mathematical biology or stochastic processes in biology. She is pretty much the only book that talks about extinction, which is from a stochastic perspective and properly. So with her guidance, I sort of jumped into this. So this was work we did before Antonio and Martin came into the picture. Extinction is certain, which for me, I don't know, I looked at that, and of course I'm not a stochastic process person, so I thought, oh God, how can we prove extinction? So for all parameters of the model, extinction is certain. Not only, so what is that? The probability to become extinct is 1. But also important is not only you have probability 1 to become extinct, but the times to extinction are finite. What about if, yes, with certainty I become extinct, but it's infinite time? Well, biology isn't going to wait infinite number of seconds, right? That wouldn't be nice. So that's one thing. But then important is, if extinction is the ultimate fate, and I'm interested in homeostasis, can I talk about before I become extinct, so the probability conditioned on not becoming extinct at time t, what is the distribution of my clonotypes? And that's called the quasi-stationary probability distribution. And one can look at 2 limits, hard niche, when as a clonotype you compete very little, and soft niche when you compete a lot. Yeah. So that's what is it that we then did? And from here, this is the reference to the work with Antonio and Martin that they pretty much set up. And to me, the beauty of this is talking to them, and because of course we all think differently, which is sort of the beauty of these things. How can this model we had applied to homeostasis? You've got your replete compartment, talk about homeostasis. But then something that hadn't occurred to me, and talking to them, we thought, wow, then we can use this for something very lovely, which is, if I've got, just imagine you are a thymocyte ready to come out of the thymus, size 1, and you're jumping into this ocean of replete cells, naive T cells. How well can you do given the properties of your T cell receptor, right? Because that's what defines you. You are a clone of size 1, you jump into this ocean. So the idea is, well, we can make use of this model with the right tools that Antonio and Martin showed me. So the idea is, as a new clonotype that is going to come into the blood and going to be trafficking in the lymph nodes, how likely are you to survive and become established as a new clone with a new molecular structure? So of course, that's going to depend on what is the peptide MHC available, how many competitors, and the characteristic size of those competitors. So if you remember, that's going to be average N, the phi, and what else? And the competition index. Yeah, those three numbers that come from the model. That's the idea. So I've explained this, and I'm just telling you that the three parameters that we've got for the proliferation rate, and of course the death, but of course for a given clonotype, we assume all cells have the same. So I've explained the model, so that's where we are, a birth and death process. And the different scenarios, and this was very important, is what can then we do depending on whether you have few competitors or many competitors. So one can actually show that this sum, when nu is very small, becomes constant. So if you compete very little, your proliferation rate doesn't depend on your size. Of course, if you are an intermediate competitor, then you have to use a full expression. Of course, with some truncation when you're doing numerics, right? Because the computer doesn't know infinity. And in the case of large number of competitors, this expression reduces to this form. So all I'm going to say is that for these two limits, the first and the third, you can do lots of things pencil and paper, not for this. So since we had never really dwelled with this, we thought, well, can we then study all the three cases? So what is it now that we want to do? So this is the scenario now. It's exactly what I've done before, but at time 0, I start with just one cell. So the idea is to analyze the behavior of this process. Yeah. And then maybe I should point out, because what they introduced me to is, of course, you look at the Kolmogorov equations, which are the time evolution of these probabilities, you cannot solve them all the time. And sometimes for the questions, even if you can solve them, you don't need such heavy-duty weapons. So in fact, I don't know who I was talking to, but, Murad yesterday. Murad, he's not here anymore, fortunately, but Murad Banaji said, well, before you start with your heavy-duty artillery, why don't you start going slowly? So that's what they taught me, these stochastic descriptors, because the time to extinction is one such stochastic descriptors, but there are many other stochastic descriptors. So let me introduce them to you. So one of them could be, if I start at size 1, what is the maximum clonal size that can be achieved by a clonotype? So if I come out of the thymus into the periphery, what is my maximum size? Not only that size, but the time to reach it. Yeah, because some clonotypes will reach a certain size, but will take forever. If it takes more than 100 years, well, we'll be dead. So it's not going to happen. So that's the idea. Establishment of the thymocyte, the naive T cell, in the periphery. And of course, that's the mathematical definition of that descriptor. Another thing one can do is, in order to reach my maximum, what is the number of proliferation events? And I know that, for example, some people who've worked on this, Rui is here, and some other friend of ours, like Alan, number of proliferation can be measured with immunology, and Sarah knows very well about one particular type of fluorescence in which you label the cells and you can track beautifully, because the intensity every time the mother cell divides halves. So proliferation events can be measured. So in order to reach the maximum, how many divisions do I require? And something important is, of course, a time is a continuous variable, but number of divisions is a discrete variable. So you want to try to develop tools for different random variables, continuous and discrete. And also, if I already have grown to a given size, what is the time into contraction onto expansion to a different size? So if I start at size I, what is the time to make it to size J? With, of course, I or J, they can be bigger or larger than that. And other descriptors that we could imagine. So this is just sort of a suite of ideas. So again, in this paper that I've mentioned, we looked at the maximum clonal size, the time to reach the maximum, time to reach a total of proliferation events, time to so many D divisions, and time to reach a given size if I come from size 1. Is that clear? So since I know you want to take the bus, I'm going to describe just one, and the rest is in the paper. And I think one shouldn't be silly and do too much, not explaining. So the maximum clonal size, so it's a discrete stochastic descriptor, and I choose this one because, I don't know, for some reason I was more familiar with continuous. So, of course, the idea, what is, we want to calculate what is the probability for my clone that the maximum is going to be 1. I start 1 and I stay 1, but I stay the maximum, or I become whatever size. Those are those probabilities we want to compute. So this is what they taught me, Martin and Antonio. To understand the distribution for this random variable, which is discrete, what I need to understand is this random variable, the times it takes to go from size I to size J. Yeah? Because if I understand this random variable, the probability for that random variable is related for the probability of the maximum being greater or equal than J. And of course, if I want to calculate the probabilities I was telling you, the probability that the maximum is J, that's what you've got to do. So you can trace it down to that. So our mathematical objective, at least for now, is to then compute these probabilities. How can you do that? Well, they introduced me to this method. Instead of dealing with the raw quantities, you use some Laplace. Martin, you're Dutch, now that you're here. Can I ask you how you pronounce that? Stiltjes. Okay, I mean, that was my guess, but I thought Martin is here, I'll ask. There's nothing better, because doesn't it sound funny when someone pronounces your name? Anyhow, that's just me. No, I've been called Molina. And for me, it's like, you don't recognize yourself. So what you can do is, instead of these quantities, and important, remember, these times need to be finite, because if infinite, then, we're not waiting for that to happen, not to see it with our eyes. It's, you introduce this variable s, and you're familiar with Laplace transform, you understand, e to the minus s, e to the minus st, with the indicator function, that needs to happen for different I and j's. And of course, in this case, I'm starting size I and I need to grow, so I is smaller than j. So what important is, of course, when s is 0, I get the probability that I'm interested. But one can do a first step argument to realize that these functions verify this very nice hierarchical structure. So if I start at size I and I want to go to j, I only am linked, of course, it's natural because it's a birth and death process, so I go one step to the right or one step to the left. So if I go one way down, of course, it's my mu, my death rate. If I go to the right, it's my birth rate, I always say the other way around, so this is a recursion in which these functions can be solved, and this is very beautiful, and the important thing is that it's a recursion, but you're only linked to one above and one below, so you can solve it like that with these g's, gk of s having this form, which again, it's a recursive relation. And of course, the first one, you know, it's mu1, because that's state 1 is only linked to 0. Yeah? So that's the idea, you do these things. And even though you still say, well, so to me this is already nice and analytical, even though you may require the computer to do it. But you know how these things behave. So let me just describe what we did numerically, well, what they did numerically. So let's just think about understanding for different values of my proliferation rate, the distribution of the maximum. So remember, this is a distribution for a clone that starts at size 1. What is the maximum going to be? In the hard niche case, so this is from the purposes of being a self-centered clone, this is the best scenario. You're out and you have 0 competitors. How large can you be? Depending on how much stimulation you can get. So of course, the colors from blue to green to red and to yellow, and I apologize if you're colorblind, that you can, so you start, if you have little proliferation, you're not going to grow very large, and as you increase, of course, the resources from your antigen presenting cells, you can grow. And you can see for the numbers we put, which were immunologically supported, you can grow to become 25 cells, which, starting from 1, that's pretty reasonable. Then and this is to me where, it was very interesting to see how now in the intermediate case, when you need to put the big expression that we hadn't been able to analyze properly before, you need to worry about, in the intermediate, what is precisely your competition index, nu, and how much signal you get. So, I should have said, so this is the histogram of X mass, X maximum, and of course, this is how large you can be. So you can imagine, of course, the more you compete, that really is, the competition index is really the driving, the most important parameter in the model. If you compete not a lot, you can just see again that you can grow big if you have sufficient stimulation, but as you start competing much more, you really, even when you have lots of stimulation, you're not going to grow very large. And then, of course, in the hard niche, when you've got lots of competitors, you're born into the world where everybody is sharing your resources, big competitors, you see that you are going to be small, 10 cells, sorry, not 10 clones, 10 cells. So what is it that, I'm not going to show more results because they're all in the paper. I wanted to give you the flavor of the tools and the methods. So what is it that then we were able to see? That for a new thymic emigrant, you basically have 2 fates. And the 2 fates are, you go outside and there's not enough signal. So no matter how many competitors, if there's not enough signal, in the short term, your probability to survive is very low. So you become extinct. If you have enough signal and this is important, and a friendly, moderate environment, there's a significant long-term survival probability, and that can be approximated by the X max, which is quite nice to see. But even in friendly environments, you never lose this bimodal structure. The idea is, if you're still alive but you're sufficiently close to 1, your chances of dying are really non-vanishing. So what is it then that this is hinting? Exactly what I just said. So this bimodal structure in the distribution of the maximum size of the clone is, in the short term, with non-negligible probability, even in friendly environments, you're going to become extinct, which makes it for these recent thymic emigrants not a good prospect. But then, if such an event doesn't occur, if you don't become extinct, The beauty of our results is that you escape extinction and it reaches the maximum size, and if it does, it's very much near the second mode of the probability mass function. So you become established at that. And what we were able to observe is that there is a low variance in those distributions, which, you know, we didn't have a reason to believe this. And to me, that's important, that the molecular properties of the TC are encoded in phi and nu, sorry, not n, because the size of the competitors, so it's really how much signal you get and how many competitors you've got. They seem to determine the clonal size, which is, oh, and that's funny because I thought I had, no, sorry, I must have, for some reason, anyhow, I would have liked to show one in which you change the number of competitors, but anyhow, you can imagine what's going to happen. The more competitors, the narrower and smaller you become. So I would like to finish, I think, with a reflection. I have to say, it's a pleasure to be here. Vijay Krishnamurthy is not here, but the support we've had, because let's start in order. We need to thank Nagasuma Chandra, because through her, you know, we've been coming to India, and in her visits, she's always been very generous, and we've met other scientists from ICTS, and I have to say, the community in India is almost like the Spanish, that, you know, your friend is somewhere else, and they say, if you're there, talk to him. So I happened to be with Suma. Sitabra Singha said, but Carmen, you should talk to Vijay at ICTS, because he's also a physicist, and he liked what you do. And he came and he just said, this was about 2 years ago, exactly. You need to organize one of these. So I have to say, it's a pleasure to have the support for all of us to be here, thanks to Suma, Shitabra, Vijay, and I really would like to say Martin, because Martin has done everything without complaining, which I regularly do. And then just going back to the science, Emily with Hugo really pushed this, Antonio and Martin for putting up with me, and I still remember Antonio, I have to say this aloud. I could never imagine that you would be here, not because this is India, but because this is so much immunology. I mean, there was one time when I thought, oh my God, I don't think they like what I'm doing, because it's not proper thing. So for me, it's a pleasure to see that you're really part of this adventure. And I also would like to, because he's gone, Robin Callard, he was with us here at a meeting in 2011, which was the first meeting we organized here with a European project with Suma and Dipankar Nandi, because he believed in mathematics. He's an immunologist who did an open degree in mathematics with the Open University at the age of 60-something. So, and he really believed that maths was making a difference. So I think all of us in the UK really should think about him. Support, support, as I said, without funding from the European Commission, these Indian adventures wouldn't have happened. And I would like to conclude with saying in Leeds, Martin, Grant and I host a Marie Curie Slodowska training network on T-cell immunology and immunotherapy. The webpage is here. We are going to, so it started exactly a year ago, and we're going to run lots of different workshops for the next 3 years with 15 PhD students all over Europe. And at the minute, if you've got a PhD in, or you're going to defend before the end of August, we've got a postdoctoral position. It's only a short one, it's only a year, and the job ad is out if you're interested to model viral dynamics of flu dip. So I guess it's thank you to Laura for talking about dips, because then you know what I'm talking about. So with this, I'll stop and take any questions. Thank you very much.